Good morning, good afternoon, everyone. Thank you so much for joining us for this session. As people are just entering the virtual platform, for those of you who've joined already, I'd really like to thank you. And I will begin just with a little bit of preamble before we enter the substantive part of this webinar. Happy New Year to you all. And I'm Marinisha. I am co-lead of Genome and Director of Ethics and Law Education at the University of Oxford. And I have the wonderful privilege of being your host for today's webinar. Just as a reminder for those of you who attended our first webinar and for those of you who are new to these sessions, Genome is a virtual public engagement platform facilitating conversations between experts and the public on developments in the field of genomics. We also hope to bring these developments into dialogue with Islamic ethics. And what we'll do is we'll tell you a little bit more about this interface between science, ethics, and Islamic ethics as the session ensues. Genome, in and of itself, is the public-facing part of the project that has been funded by the Qatar National Research Fund, Genomics, Islamic Ethics and Public Engagement. And through this project, we will be exploring how Muslim public awareness of and engagement with genomics can be improved. Today is the second in this series of public engagement sessions with experts and yourselves. And if you missed the first one, a brief summary is that we covered at an introductory level the role and importance of public engagement in shaping science, why you and your views matter. And if you missed it and you'd like to catch up, then please do get in touch. We can make the recording available to you. The focus of today's webinar is very much building on that first session. And what we'll do is we'll delve into more detail about genomics as a field, its history, the science, looking more broadly at secular ethics and how both the science and the ethics interface with this domain of Islamic ethics. I will introduce the speakers and the format of the sessions shortly. But what I do want to do is cover a little bit of housekeeping with you. I'm really sort of looking at the number of attendees, and that number is going up gradually. And although as attendees you won't be visible and you won't be able to directly engage with us, there is the Q&A and the chat function, which will allow you to put questions in, respond to the speakers, express your enthusiasm for what is being said, or even disagreements, or to embellish it with your own expertise and your experience. When you're putting in questions in the chat, please do introduce yourselves. We'd love to know who you are, what you do, where you're from, and then put your questions in the chat. There's also ways in which you can engage with Genome as a project, so do join our mailing list, email us. There's a Twitter handle for the event that Muna will be pasting into the chat very shortly. The hashtag for today's session is #Genome, so please do tweet as we go, and do join our Twitter handle as you see in the chat. The format of the webinar, most of you will be very familiar with the virtual platform. What I will do is introduce each of the speakers in turn. They will have 20 minutes to present using slides and narrative. After the 20 minutes, you will have the opportunity to engage with speakers directly with the questions that you put into the chat and the Q&A function. And you'll have 5 to 10 minutes to do that. Once we finish with all the speakers and the individual sessions, we will have 30 minutes for plenary. And what I want to do is use that opportunity to have the speakers in dialogue with one another. So our panelists will have the opportunity to reflect on one another's presentations and also ask questions. And then I will open up to our attendees to also ask additional questions and put in any additional remarks. Without further ado, I do want to open up the session and introduce our first speaker, who will be telling us about genomics as a field of scientific inquiry, and who'll be covering the science and the history. It's my wonderful privilege to introduce Professor George Nummer, who is a professor and coordinator of genomics and precision medicine at the College of Health and Life Sciences at Hamad bin Khalifa University. And George is renowned for many achievements in research and in genomics, one of which was to develop the first mouse model of human cardiac congenital malformations as part of his PhD. And it's one of the most cited pieces of research in the field. It's one that I'm particularly interested in. So thank you so much, George, for joining us. It's a wonderful privilege to have you, and I will hand over to you now.
Right. Thank you, Marinisha, for this excellent introduction. I would like first to thank Dr. Muhammad Ghali, who is really instrumental in putting this Genome project. And of course, you know, we've been battling throughout the last 2 years with, again, something related to genomic, which is SARS-CoV-2, which in fact, because of all the genomic information we gathered throughout the years and throughout the centuries, we were better equipped to fight it. And I'm going to share with you some of the knowledge that we gained throughout history and what are the key events that have shaped our understanding of genetics and genomics since the day we started, you know, understanding that the genes are the key elements in regulating our daily lives as human beings, and of course, as other species around us in this universe. So with no further delay, I will share with you my presentation, and I will go through the main events that have shaped history and especially humanity in terms of our understanding of genomics and genetics. So my presentation will start with a disclosure, of course. I hope that everybody can see it. So okay, so I'll start with, again, acknowledging the importance of sharing knowledge. And this is what Dr. Ghali has really put into context while starting this webinar series last year. And today, what I'm going to really talk about is the history about the genetics and genomic field. Probably, you know, when I was looking in the in Google, as we always do nowadays in this era of globalization, is to find, you know, key events and then try to share them with everybody. And of course, giving our personal flavor in terms of giving history another look while sharing knowledge. And I found this really interesting map of all the historical background, and I saw it from a website referred to as Living DNA. And in fact, it was a history of DNA and genomics throughout the years. I tried to dissect it and show how important, you know, the last century was for our understanding in genomics. So with no further delay, I would like to start with the key concept of genetics and genomics. And again, if you go back into history, like 4,000 and 5,000 years BC, you can see that even, you know, at the peasant level, where people use agriculture as a tool to survive in terms of humanity, early farmers begin selective breeding. At that time, you know, there's nothing known about genetics. We did not know anything. But it took years before scientists, before we didn't have scientists, we have philosophers. And that's where the Greek philosophers were important, like Aristotle, to put the concept of inheritance. And in fact, this dogma of inheritance have made it a real influence with Gregor Mendel. Everybody in the field of genetics, we took courses of genetics when we were at school. We remember what is the importance of the experiment done by Gregor Mendel. And this is back in 1857, where he discovered that pea plants, you know, can have different shapes, can have different attitudes or environmental reactions. By just crossing them, he could come up with dominant or recessive traits that have been favored in terms of surviving harsh conditions and giving the best products. So the notion of inheritance was also at the same time dealt with a different attitude, probably from Charles Darwin, where he put the origin of species. But at the same time, the concept of inheritance was a key concept that geneticists developed throughout the years. And here, I don't want to delve into the aspect in terms of culture or religion, but I'm stressing upon the point of the concept of inheritance from one generation to another, whereby some genetic information is shared that is going to dictate some of the elements of survival or physiological action that most humans share between them and most species shared between them. The major discovery that really shape up the knowledge in terms of inheritance was the discovery of DNA as a substance. And again, in fact, if you think about it, the first isolation attempt of the DNA material is practically 150 years old. Back into Friedrich Mitscher in 1869, where he discovered the substance that we referred to as nuclein at that time, and that nowadays is referred to as DNA. And later on, the researchers in the field try to shape up the knowledge into what we know today as chromosome, and of course, how genes are now set into chromosome. However, the real breakthrough in terms of our knowledge of genomics came from the 20th century, where essential knowledge started with the actual discovery of the composition of these DNA molecules in terms of nucleotide, adenine, guanine, cytosine, and thymine that we know today. And this moiety, which is linked to a sugar and phosphate that made the backbone of DNA, was really put into real action that we can visualize. Again, because of the discovery of the double helix structure of the DNA, which was made available because of the growing technology at that time, and which were focusing more on X-ray diffraction that have helped visualize the DNA in the first time. However, most of our understanding of genomics and its relation to disease that affect the humans, and this is the challenge that I'm going to stress upon in this seminar, is that genomics is being used to address the challenge for a human, which are diseases, and not only traits that we are going to acquire from generation to another. And this is where ethical issues arise. In the past, the ethical issues, as I'm going to describe them, were related to the concept, the dogma, but later on, they were related to technologies and the use and abuse of technologies. So after describing these moments, I'm going to put the challenges that faces humans in terms of apprehending the notions, but most importantly, and the use and abuse of these technologies. So after discovering the double helix structure of the DNA, and of course what it made as a breakthrough, the other major breakthrough came from sequencing of the DNA. And this started back in the 1960s and 70s, and mainly in 1977 with the discovery of Sanger sequencing, which has helped identify several genes that were implicated in several diseases. But again, that was tedious, that was really very, very time-consuming, and it took many years for scientists to come with a semi-automated or automated approach to do DNA sequencing and to get to the first draft of the human genome late in the 1990s and early 2000s. And again, during that period, from 1970s till 2000, many things happened at the level of our apprehension of technologies and the use and abuse of this technology. And people were drifted into doing things that probably will not help scientists, but, you know, add more curiosity to how we can handle genomic. And this is how Dolly, the first chip, was cloned in 1997, even before we know what composes the human genome, and that the first draft of the human genome was sequenced in 2000 and 2001. So telling you again that, you know, key events are driving humanity, but more importantly, that guidance should be done throughout these events to really focus on what is more important for us, which is to save human lives. And in fact, this is where the focus I'm getting into it, and I'm lucky to be also in an institution where, you know, precision medicine is the focus, and this is where the use of genomics is to help people get better. And the terms precision medicine took a big shape after the Human Genome Sequence project was done. And then, you know, other studies have been in the market, mainly the Genomic-Wide Association study that helped correlate traits of different diseases with the makeup of our genomic entities. And this has been even more put into practice with the discovery of editing tools that we refer to as genomic editing tools, like the CRISPR discovery in 2012, which has been used nowadays in terms of trying to cure diseases. And this is the know-how that is being transformed into application that's going to serve the suffering humanities. What I would like to conclude with this part of the talk is that the challenges that face humanities at this point in terms of discovery of genomic and genetics were related mainly to these 4 points. First of all, the challenges were related to the acceptance by societies and, I will say, rulers, because the rulers in terms of politics were the ones that are governing these entities. The second is the limited technologies that have faced most of the researchers to come up with new knowledge. And that's why you can see that during the 20th and 21st century, all of this were accelerated, whereas before the 20th century, all was limited because of limitation of technology. Now, the other major issues that were for scientists, for societies, for human beings, were the ethical use of animal models in order to advance our knowledge in terms of genetics and genomics. And more importantly, are the ethical human concerns that are affecting our daily lives. And I will dwell on it when I'm going to expose the explosion of our knowledge in terms of genomics, which was related to the increase in knowledge in technologies. With no further delay, I will expose briefly all the main events that have happened in terms of the history of genetic engineering technologies. And these are listed here just to give you insight about how these technologies have shaped our understanding of genetics and genomics. I'm going to skip some of them due to lack of time, but I'm going to emphasize on the most important features. Again, the best advancement that happened during the 20th century is the double helix structure of the DNA. And that was really a big explosion in terms of genetic and genomic information. However, that came with some problems, because the technology that was used is the X-ray diffraction. And the X-ray diffraction, as you might know, with all X-ray technologies, is that you are exposing the humans or the researchers to these sort of carcinogens, okay, because they are going to have an impact on our own DNA when you are exposed to X-ray. So see how the challenges for scientists was really limited because we cannot have additional technologies. Now, what happened after that is that DNA was first made in a test tube for the first time 5 years, down the discovery of the double helix structure, and additional technologies were discovered. Again, the use of cloning DNA came because of major discoveries that made in terms of bacterial genome, whereby we discovered the restriction enzymes that were used to clone the DNA. And then if you go to the first transgenic animal, it was done in 1981, where scientists used to put a small fragment of DNA from rabbit and put it inside the mouse. And that was the first transgenic animal that was what formed. And, if you look at it technically, it sounds nowadays that, wow, this is easy, but it took years to come up with that. So the major discoveries that came because of the understanding of recombinant DNA technology was important for improving human health. And again, here we are talking about diabetes. The first genetic engineer human drug, which is insulin, was done in 1982. It's not that far away, but again, it is because of our knowledge of genetics and genomics in humans and in other species, especially bacteria, that really made the differences. Again, I will stop from enumerating all of them, but I would like to put the PCR, because we are using now PCR to screen. PCR was first discovered in 1983, and it was put into the actual work because scientists were really obsessed with the idea of working with DNA in test tubes, okay, and visualize DNA, and this is how it came to life. So I will skip some of the others technology to come to the real one that I would like to highlight here is the discovery of the principles of gene editing. And again, while getting the Nobel Prize for CRISPR was last year or 2 years ago in 2020, the discovery of the principles date back to 1993, but it took years for researchers to come up with a really good formula to understand the basis of CRISPR and why it is useful to cure diseases, as we are going to see. Other attempts, in terms of DNA editing and work with cells failed in terms of our apprehension of diseases. I will mention only the RNA inhibition that was also awarded a Nobel Prize in 2004 or 2005, but didn't help us in our daily life in terms of curing diseases. I mentioned the first next generation sequencing as the biggest investment in order to understand the genomics and traits that are linked to diseases, and it really accelerated the way we are using genomics now in our daily lives. I will put the identification of stem cells and using of the first induced pluripotent stem cells as an attempt to use also genetic information in a context whereby each individual will be using his or her own cells to cure him or herself, whether there is a disease or not. So these attempts, have been available because, again, of our increasing knowledge in terms of human genome. Now, what I would be ending in terms of history is to say that the first human trial for CRISPR were approved in 2018, and I shall mention that the FDA approved the first test to correct a genetic defect using CRISPR and to cure sickle cell disease, and that was approved, last year, practically in March 2021. What were the challenges throughout these technologies? Of course, what scientists or not scientists would be eager to do is to abusive use of technologies for their own curiosity, for their own projects, for material gains, etc. The second is the unethical experiments that could be conducted even on humans. And again, I failed to mention probably in the history, and I'm going to highlight it here, is that the first trial using CRISPR were done on human embryos in 2015 in China, but they were not approved ethically. And that created really a big challenge for humanity, not only at the level of the country, but everywhere in the world. Again, that was also the issue cloning Dolly and trying or attempting to clone other species, including humans. So that were really major unethical experiments that were done and that were curbed, luckily, by some legislation that I'm going to mention at the end of this talk. And the concern of sharing genomic samples and data was really the biggest challenge in the last 10 to 15 years. And that is not related to the fear, but practically, again, for the abuse of having some people, getting these data and manipulating them. And finally, I want to mention the inequity in sharing technologies and products, which was really favoring advanced countries versus, developing countries. And that lead to really I will say it, unequal sharing of knowledge, which is really a major threat to the survival of humans. I guess, we are exceeding time, but I want to conclude now. And concluding is, learning. So what I am going to summarize the historical background of genetics and genomics is that we want to highlight history and historical facts to provide positive feedbacks, okay? We want to learn. We don't want to repeat mistakes, and we want to increase our knowledge, but this increase in our knowledge should be really focusing into improving and preserving human health. And this should be the focus in genomic studies. And more importantly, we would like to have appropriate legislation to protect humanity and to share knowledge to preserve human race. With this, I'm ending this talk and thanking you again for listening, and I'm happy to answer any question during the Q&A and to discuss all the pertaining information with the other panelists. Thank you.
Thank you so much, George. I mean, that is a fabulous coverage of the history of genetics and genomics from Gregor Mendel to Jennifer Doudna. And you've put so succinctly this transition from us learning about the science for the science's sake to really thinking about how we can apply that knowledge for the betterment of human health. I was wondering if I could take chair's prerogative and ask you a couple of questions. The first is around whether you'd be able to tell us a little bit about the work that you do on a congenital heart disease to bring to life the practical applications for a lot of the types of knowledge and the technologies that you've introduced for us.
Yeah, I'm happy because this is something that I cherish a lot. I've been spending more than maybe 25 years since I started my PhD working on the heart. Okay, and why we are working on the heart? The heart is the first organ to be formed, and any defect in the heart will lead to embryonic lethality. And in fact, again, why I choose that, it is because it is going to help alleviate most of the problems that newborns are facing. Congenital heart disease account for 1 case for each 100 newborn in all the countries, which is really a major threat. And the first year of life is threatened by lethality or death because of congenital heart diseases. And that's why, I took it to my heart and to the heart of many of the researchers to work on the field to understand better what are the genetic background for this disease. And when I was a PhD student, as you mentioned, my hallmark discovery was really to unveil the mutation in the gene TBX5 that causes Holt-Oram syndrome, which is a very, very recessive mutation that causes a part of the congenital heart malformation, but also limb defects in neonates. And we were the first to introduce a model in mice to mimic exactly what we have in humans. And that was good because you establish a model whereby you can use it either for early diagnosis or for understanding the mechanisms or to cure. Because we know that the only cure now for congenital heart disease is mostly either giving some drugs, okay, which is going to be long taking all their lives, the children who are affected till adulthood, or performing surgeries. And with surgeries, again, you have risk of many problems, although the surgeries are done well. So after discovering the genes, the other thing was challenging for us is to discover that what are other genes that are implicated. And to do so, we need the participation of the communities. And this is how I established in Lebanon the first congenital heart disease genetic program in the region, where we recruited more than 1,000 patients and the family members and start screening to see what are the genetic defects. Fortunately or unfortunately, we only, unveiled only 5% of the genetic background or the genetic mutations. And this is challenging still for us and for many in the field, because it seems that it is not only one gene that can be mutated that causes congenital heart disease. It is a network of genes. And that's why it is difficult to come up with a complete solution, but at least we are on the right track and we know how to be important in offering genetic counseling to the parents who are having problems when they have children with congenital heart disease, because they will ask you, well, is my second child going to have congenital heart disease? You need to have the answer genetically, and that we are trying to provide now with a good accuracy.
Thank you so much, George. I mean, that's incredibly important. Towards the end of the session, we'll come back to this question of how an understanding of genetic mutations can lend themselves to therapeutic solutions and the sorts of challenges around that and the promises of genomics around that. But what I wanted to do was reflect a little bit on what you very helpfully identified as the sorts of challenges around genetics and genomics research, these issues around abuse of technologies, unethical experiments, the challenges around sharing of samples and inequities in access and the sharing of technologies. And because this program that we're running is very much about public engagement, I wanted to ask you, as an expert in the field, what do you think is the role of the public in aiding an understanding of some of these challenges, but also helping us address them?
Yeah, I think the input from the public is very important in terms of directing the research in one area or the other. Now, our role is to share, okay? And that's why I say it should start from us. We need to share knowledge with the audience, with the public. Otherwise, we cannot get good feedback. The feedback that we are going to get is to reflect on what is needed from the public. It will really direct our research to the needs. And this is what we want to have. We don't want to only do research just for the fun of doing research, but to help the need, to help cover the need, and of course, alleviate the burden on the suffering families, especially when you have congenital diseases or common diseases.
Brilliant. Thank you so much, George. What a really positive note to end your part of the session on. That there's this need for dialogue between scientists and the public for us to be able to map need and unmet need. Thank you so much, George. Thank you. And on that, what I will do is I'll move on to our next speaker. it's my wonderful privilege to introduce Dr. Tara Clancey, who is a consultant genetic counsellor and honorary senior lecturer in medical genetics, and her main clinical and research interests are cancer genetics and the ethical, legal and psychosocial issues in genetics. And what Tara will be doing for us, which is incredibly helpful, building on George's session about the science and the history, as well as some of his expertise around pediatric genetic diseases. Tara will be giving us an overview of some of the secular ethical implications of genomics before we go on to the Islamic ethical issues. Tara is a renowned expert. She runs several groups in the UK, including the Gen Ethics Club, as well as the UK Fetal Genomics Group. And I've had the wonderful privilege of serving with Tara as a member of the Nuffield Council on Bioethics. Thank you so much, Tara. It's wonderful of you to join us, and I'll hand over to you now.
Thank you. I always worry about the technical issues here, but I think this is working. Okay, so I've sort of decided, having talked to Marinisha earlier in the week or last week, to focus down on some of the ethical issues in pediatric genetics and genomics. And even then, I'm only going to be kind of scratching the surface. And also say thank you for a very generous introduction as well. So the outline of my talk, I'm going to talk briefly about genomic medicine services and genetic counselling in the UK, really to put in the context where I've worked and how our services run here. There are other models, clearly, but I thought that might be helpful. Obviously, I want to talk a bit about ethics in a more general sense, ethical principles in healthcare, and because I'm focusing on paediatrics, talk about the concept of the best interest of the child. And then I'm going to look at three areas to do with paediatric genetics and genomics, about tumour testing, whole genome sequencing of newborn babies, and genetic testing in childhood. It's going to be a bit of a whirlwind tour of all this, I suspect. So genomic services in the UK, there are, I was going to tell you the number, but I can't actually remember. It's sort of under 20, so we're based in regions around the UK. So that includes England, Scotland, Wales and Northern Ireland. And genetic diagnosis, testing and counselling for rare conditions. Now, the conditions individually are rare, but there are many of these conditions, and most of this work is undertaken by a multidisciplinary team, which includes, but isn't limited to, genetic counsellors such as myself, doctors, particularly in clinical genetics and metabolic medicine, and clinical scientists. And as people are more familiar with the area might will already know, both technological developments, which George has gone into earlier, and the complexity of inherited diseases requires inputs from other specialities. So genomics in the UK, as in many other countries, is becoming part of mainstream medicine and healthcare. And then to think about genetic counselling in the UK, this is almost exclusively provided by genetic counsellors and doctors, and we have specialist education expertise in genetics, genomics and counselling. And so the sorts of things we do, I think most of you will be very familiar with. So, for example, assessment and interpretation of personal family history, counselling to support individuals and families both understand and learn to live with their genetic condition, interpret and explain complex, incidental and or uncertain genomic information. And also, one of the things I'm very interested in, addressing the ethical challenges that arise. And in this context, it links to what George was saying, but I guess, so thinking about unethical experiments or unethical use of technologies, but I guess in the clinical context, it's more about thinking about the sort of services we provide and the decisions patients might make about having or not having genomic tests, and also, for example, disclosing and sharing genomic information. I think it's perhaps helpful to think about, you know, well, what does ethics mean? And it means many things to many people, I guess. So we could think about it as doing the right thing. We could think of it as knowing what the right thing to do is, which may not be the same as doing the right thing, I suppose. It could be something broader about how we live our lives, how we make decisions, and it could also be how we take account of others' interests and needs. So ethics derives from the Greek word ethos, which means a way of living. So it's really concerned about human conduct, such as the behaviour of individuals in society. It looks at examining the justifications we have for our judgments, so what is right and wrong, what is just and unjust. And it reflects on us and our interactions with others and with the world. So what is ethics about? Now, this is my view. Other people who are part of this seminar may or may not agree with some or all of this. So I guess I see it as it's really a fundamental part of being human. It's not an optional extra. It's about considering the impact we have on our responsibilities towards others. So that might be people that we know and we meet, but also people we do not know and will never meet. And I guess for me, looking at it this way, ethics is about values such as respect, trustworthiness, responsibility, fairness and caring. And values such as these are necessary for us to be the best we can be, both as individuals, also as part of our family and friendship networks, as professionals, as colleagues and employees, and also as citizens. So that might be locally, so for me in Manchester, for example, nationally in the UK, and globally. So there are very many ethical and philosophical theories about ethics, and I'm going to focus. I don't think these are the only answers, but they're widely used. I think it's helpful to talk about the 4 ethical principles in healthcare. So one is autonomy, which is about respecting individuals' abilities to make and act on their own informed decision. Another is justice, again really refers back to George's presentation. So I think about equitable decision making and equitable use and distribution of resources, for example. Beneficence, which is about doing good or doing the best for others, and certainly hopefully ensuring that any benefits outweigh harms. And then non-maleficence, which is a bit of a mouthful, or about doing no harm. So I think there were quite a lot of strengths about the 4 principles. One is that they could be considered to be an intercultural approach to ethics. Now, I have put a question mark there because there will be some dispute about this. But one of the things I think that's quite interesting to consider is that rights and duties or obligations are defined in the world's major religions and in secular traditions. Although, of course, what these rights and duties or obligations are and the balance between them can and does differ. And it's also, I think, interesting that beneficence and non-maleficence both have their roots in ancient Eastern and Western philosophy. Another strength of the 4 principles is that they're applicable in a very wide range of situations. And I guess the other important thing they do is they give us a framework or language to express and explore conflicting views. I think equally it's important to be aware about what the limitations are. So it may be perhaps all these 4 principles may not apply in all situations. Another which is more challenging, I suppose, or challenging in a different way, is do the principles, if we're applying them, do they point to the same outcome? I think another big issue is that sometimes, certainly for those of us in clinical practice, there can be a bit of a temptation to use the 4 principles, what we would call it here, as a box ticking exercise. You know, I've thought about autonomy, I've thought about justice, I've thought about beneficence, and I've thought about non-maleficence, and kind of that's as far as it goes. And we need to think about other important values such as truth-telling, transparency, trustworthiness, and very importantly, showing respect for patients and families and their values. So I guess I think this shows that the 4 principles are the beginning and not the end of ethical deliberation, but they're certainly a very good starting point. So the reason I'm going to be talking about best interests of the child is because it's a phrase that's commonly used, certainly in the UK, and it's a useful concept, but it's also it's not necessarily a concept that's very easy to put your finger on. It dates back to, in fact, almost 100 years, when the League of Nations, which was the predecessor of the United Nations, set up after the First World War, adopted the Geneva Declaration. And that's the first recognition and affirmation of rights that are specific to children, and also the responsibility of adults towards children. There were, in 1959 then, there was the United Nations Declaration on the Rights of the Child, and then 30 years after that, the United Nations Convention on the Rights of the Child. And the best interest of the child is one of the core rights. It's quite difficult to define what the best interests of the child are. I guess one key thing that is really important for those of us in clinical practice to remember, it is above and beyond, wider than best medical interests. This is what the General Medical Council in the UK would want us to include. So it's the views of the child as the child can express them, the views of the parents, the views of others who are close to the child, which might be other family members, the cultural, religious and or other beliefs and values of the child and parents, the views of the health professionals involved in providing care and of other professionals who've got an interest in the welfare of the child, and also thinking about the choice, if there is more than one choice, that is least restrictive of the child's future options. So I'm going to move on to something that's a bit more specific to genomics now, and think about ethical issues in paediatric cancer genetic testing. So personalised medicine and targeted treatments based on specific genomic variants can be really helpful in providing information about the likely cause of the condition or the cancer, and or about treatment options. And screening for these genomic variants is now part of standard diagnostic workup of cancer diagnosis in many settings. I think at the same time we have to be aware that patients and families and clinicians, and I would include people working in genomics as well as, for example, oncology, can have high, often unrealistic hopes from genomic testing and also personalised medicine. And I think phrases like personalised medicine and precision medicine can kind of contribute to some of those, the kind of disconnect between what people kind of hope and expect and what can be delivered. Another difficulty is that tumour testing is often not seen as genetic testing in the way that testing DNA from a blood sample is. So genomic variants that might be identified in tumours may originate as germline variants. In other words, they may be present in all cells, not just in the tumour. And if there is a germline variant, then the child's parents, siblings and so on may be at risk of an inherited cancer condition. And genes on panels that use genomic testing often differ depending on the type of cancer, but they often do not differ for paediatric and adult settings. So I think what we need to understand, and I don't think this is surprising, it'd be surprising if it wasn't the case, parents are often motivated to seek tests, even if there's a very small chance that these will benefit the child. And if there's a genomic variant that's found and it suggests a germline genetic cancer condition, the parents, clinicians and the child, depending on the child's age, I guess, may be unprepared for this. And one of the things that it's important to know is that germline variants account for around 10% of childhood tumours. So if there was no explicit consent for tumour testing, or if the child, again depending on the child's age, and parents were told the information to get more information about the cancer, then finding a possible germline variant can lead to distrust or mistrust. If a germline variant is likely to be associated with the child's cancer, we can discuss confirmatory testing. And the sorts of problems that can come up then is, well, what if one parent wants this but the other doesn't? And what if the child disagrees with their parents? Another question is, well, what if the variant is actually associated with an adult onset cancer? So that might be something that's perhaps more an incidental or secondary finding. So confirmatory testing might not benefit the child at this stage, but might benefit the child's adult relatives. So then to move swiftly on to newborn screening and whole genome sequencing of newborn babies. And again, this is just, I'm setting it in the context of the UK, because obviously that's what I'm most familiar with, and I'm aware they'll be different in different settings. So newborn screening here screens babies for a number of relatively common preventable or treatable early onset conditions. In addition to that, parents then have the choice to use information in or prior to future pregnancies. So for example, they may decide not to have more children, they may decide to consider prenatal diagnosis or pre-implantation genetic diagnosis. Now, whole genome sequencing offers the possibility of screening newborn babies for additional, even rarer, preventable or treatable early onset conditions. It could also screen for early onset conditions which cannot be treated or prevented currently. And that has an advantage of avoiding something that we often call the diagnostic odyssey. So what can be a very long delay from the first concerns about a baby to a diagnosis being made. It also opens up the possibility of clinical trials for very rare conditions. In addition, whole genome sequencing might identify later or adult onset conditions that might affect the child when he or she is older, but also relatives, and might also identify carrier status that is largely only relevant to the baby's future children. So to think about some of the potential problems, I guess it's not the only one, but a really important one is that interpreting these results in a baby who is well or apparently well can be difficult and misleading. So whole genome sequencing generates lots of data. Generating data is the first step. Interpreting, understanding that is different and time consuming. Whole genome sequencing may end a diagnostic odyssey on a child who becomes unwell, but it may also begin a new odyssey. So there can be false positive results and uncertain results. Babies may be treated unless they're inappropriately. Importantly, the result may not distinguish between early and later onset versions of the same condition. Parents may experience worry and anxiety, and there's a concern the parent-baby bond may be affected. And screening for later and adult onset condition results may affect expectations about the baby, for example, and it removes the baby's choice to know or not to know about these later onset conditions when they're older. And I guess there's also a concern that whole genome sequencing may negatively affect individual society's attitudes to difference and disability if, for example, people decide not to have whole genome sequencing or not to act on that result in future pregnancy, for example. And I was trying to time myself, apologies, I think I'm still within time. I'm talking about genetic testing in childhood in a more general sense now, and really this is when there's a known family history but the child is currently well. That's what I'm going to focus on. And there's a huge amount of guidance, some from the UK, some European, but also international guidance. And certainly this tends to focus on the child's right to make decisions and the impact on the child's future autonomy. So I guess key to this would be regarded having the child's best interests at heart. And largely where testing is about future risk, as a child is well now, or might be about carrier testing, then the general recommendation is to delay testing until the child can decide when and if to be tested. Because testing in childhood removes the opportunity of the child to make their own choice. And then if there's a known family history of a later onset condition, the general recommendation is this should only happen if there's a specific reason not to wait until a child's older and could be part of the decision-making process, for example. Now, I guess some things we have to, or I want to acknowledge here, is it's difficult to determine psychosocial benefits and harms to the child, parents and families of testing and of not testing. So some of the questions to consider is how old is the child? So it may be, and it's not that we could say every 5-year-old can make certain decisions, every 10-year-old can make other ones. But how old the child is important. And what are the implications of genetic information at different ages, or probably more relevantly to different developmental stages? What about having a genetic test now versus learning about the family history and the genetic condition gradually over time in an age-appropriate way? I think it's really important that we think about what the parents have identified as the benefits and risks of testing now. And I guess, sorry, that was my phone telling my time's up. And then what benefits and risks the child has identified. So this is my last slide. So if we think, and this isn't just applied to genetics and genomics, so how can we discuss and resolve ethical dilemmas? I think key for me, people who know me well might be surprised that I say this when I think about my views, but key to this is on the focus should really be on reaching the best decision, not on winning the argument. That's quite a lot to ask of ourselves sometimes. I think it's about practicing humility and being open to other people's views and values, reflecting and being aware of our own beliefs and values, and recognising we all have ethical blind spots and cognitive biases. And I think then I'd also make a plea that we seek appropriate support, and that should be people who we trust, but also not just going to people who are going to give us the answer that we want. So that's my last slide, and I'd be happy to take questions now or later. Thank you very much.
Thank you so much, Tara. That was incredibly helpful. What a comprehensive summary of the sorts of ethical issues that arise within pediatric genetics and genomics. And I had a few sort of a combination of questions around clarification, but also an opportunity for us to spend a bit more time on the sorts of issues that you've introduced, if that's okay, Tara.
Sure.
The first is, you mentioned how genetic testing within pediatrics can lead to high or unrealistic hopes and expectations. And I was just wondering whether you'd be able to tell us a little bit more about what that means, and particularly what it means in practice, particularly given your expertise and your experience around genetic counselling.
I think it's I guess what's difficult, and I think those working in genetics and genomics are part of the problem. Hopefully we're part of the solution as well. But it's sometimes it's about the very language used about genetics and genomics. So the blueprint, the key to life, and it's not that I don't think those phrases have some kind of degree of objective truth, but I think it can make it seem as though genetics and genomics have the answer to everything, I think. And I guess in the context, not just of paediatrics, but it's perhaps even more acute in paediatrics, particularly if parents have sick children, or if you're sick yourself, or there's someone you're close to, you care about. There is hope. It's a fundamental part of human nature, I suppose, that you want there to be a cure, you expect there to be a cure or some sort of treatment. And I think it's one of the reasons that talking about personalised medicine and precision medicine, it kind of sets itself up, or it's being set up as though that that there is an answer, and there will be a treatment available if we only look hard enough. And I guess it's a balance, because I think it's, hope is really important. I think, say, when Pandora's box was opened, the last thing left was hope. But I guess it's about how we get that balance of being realistic about things, and that can shift quite rapidly. So sometimes it's about education, but I certainly don't. I mean, sometimes I think people, first I was working in genetics and genomics, can kind of feel if only people understood it more, they'd stop having these unrealistic expectations. I don't think that's entirely true. I don't think it's just about giving a better and better explanation of genomic variants. And I guess it's about kind of sometimes having a dialogue about what people's getting it up front, what people are hoping for and expecting, and then you can discuss from a clinical point of view what you can actually deliver. Whether or not that's answered your question, I'm not sure. Sorry.
No, absolutely, Tara, and you very nicely sort of presented a segue into my next question. One of the things that you very helpfully did in your presentation is give us an overview of the sorts of ethical commitments, the sorts of ethical issues we'd want to be thinking about within this sort of the arena of pediatric genetics, particularly around autonomy and consent. And you raised this issue of the challenge around not explicitly getting permission or consent around tumor testing. And I was just wondering whether you'd be able to tell us a little bit more about what you meant by that. You know, are there cases, are there examples that you can share with us, and you know how that then leads to this issue of mistrust that you flagged for us?
Well, I think it's probably that. I mean, for example, I don't know if I had a bit of me chopped out or off, I wouldn't really know what was going to happen to it in the pathology lab. And to be quite honest, I would probably rather not know some of it. I mean, I wouldn't feel I need to know. I guess it's the trust issue is about that you're trusting people to that both their expertise, but also that they behave in an ethical way. And I think sometimes people work in clinical settings, because we can so obviously see the benefits of looking for or looking at, we don't always think that, first of all, there might be downsides, or even that patients might not know we're doing it. I mean, sometimes the converse is true, and when we're talking about genetic testing, people think we're going to do a lot more with it than we are going to. So I think it's one of the difficult things, it goes back to the thing about being human and being ill, or people you love and you care about being ill. It's quite hard to remember everything that's discussed then. And I think that sometimes it's the sort of language that we used, and we can retreat into using increasingly technical language, which in an effort to explain things, whereas I'm a great believer in what the phrase I would use, people getting the gist of it. They don't need to know. So it's a bit like, you know, when someone comes around to fix my boiler, I'm really pleased you can sort it out. I don't really need to know how you're doing it in any great detail. That's the way that I would see it. You know, equally, I don't mean that if patients, children, families have questions, you know, I would go with that. I guess it's about being led by the patient, I suppose.
That's really helpful, Tara. And, you know, I am abusing chair's prerogative, but I think it really is a nice way for us to sort of connect to the next part of the program. You said very aptly that a lot of this isn't necessarily just about patients, families, the public getting more knowledge. Some of it is also about dialogue. And I was just wondering, you know, for our audience, given that this project is very much about public engagement, could you tell us a little bit about the role of public engagement in the UK and your experience of it, and what role it plays in that sort of trust building, that dialogue building you described, or what sort of role it should play?
It's probably easier to talk about what role it should play rather than what sort of role it does play. I mean, you might remember I said about how the 4 principles can be used as a box ticking exercise. I think sometimes, in all honesty, public engagement can, or it's, I guess the phrase we would use, it's being paid lip service to. I think when it's done well, it's fantastic. It's not always done as well as it could be. So one that's been done recently, as people might have gathered, I have some reservations about whole genome sequencing in newborns for various issues, including appropriate use of resources, as well as the ones I outlined. But there was a really fantastic piece of public engagement work done around that in the UK. And I think that's a really good example of how to do it. And I think it's because when you're embedded in your work in a particular area, so for me it's been genetics and genomics for a very long time, for all that I think I'm part of, I think I am part of wider society, when you have the expertise from being embedded in it, but sometimes you can't see the broader issues. And one of the great things about, I mean, the public is too broad a term. There's different sorts of publics. So there's the public who have a personal vested interest because they may have a family history, for example, and you know, and the public more generally. So there's more than one type of public, I suppose. But either because of people's lived experience of a genetic condition, or because of people's interest and curiosity, the public in that sense will ask questions that won't occur to us because we're so focused. We don't see a wider picture. We can see a very in-depth but quite focused version of what the issues are. And it's really interesting when you have somebody who's new to it or naive to it or coming from a different perspective, and you can be thoughts of it like that. And I think it's almost like, I'd call it as like a light bulb. And in hindsight, you might think, of course, we should be thinking about that sort of issue. And I guess it's also, you know, a lot of the work, and I think it's probably the same with the work in Qatar. It's done on the basis of public funding. And if we want to continue, almost like a kind of self-interest as professionals, if you want to continue, then you've got to show that what you're doing is delivering public good.
Absolutely. I mean, that's incredibly helpful and a really important note to sort of end your presentation, Tara. I mean, you provided, you know, an expansive moral universe of the sorts of ethical issues, the responsibilities, the obligations around pediatric genetics. So I just want to thank you so much. And I'm sure the other panelists, as well as our audience, will be picking up on some of the issues that you've raised, particularly about the role of the public and involvement of public in governance and determining what the responsibilities are around genetics and genomics. Thank you so much, Tara. Thank you. And with that, it's my wonderful privilege and honor to introduce, last but not least, our third speaker, Professor Mohammed Ghali, who is my teacher and my mentor, and is the lead PI of the project within which Genome is situated. For those of you who don't know, Professor Ghali is the Professor of Islam and Biomedical Ethics at the Research Center for Islamic Legislation Ethics, and his research interests are vast, but they lie at the intersection of Islamic ethics and biomedical sciences, and he's also the editor-in-chief of the Journal of Islamic Ethics. And within the remit of Dr. Ghali's numerous publications, there's policy work around population genomics, as well as really important work around Islamic ethical issues relating to disabilities, which I think also connects with some of our conversations today and the sorts of questions that both Tara and George raised earlier. So without further ado, Professor Ghali, I will hand over to you to tell us a little bit about Islamic ethics and genomics and the sorts of historical questions and contemporary challenges that you are grappling with. Thank you so much, Professor Ghali.
Assalamu alaikum, everybody. Good afternoon. Thanks, Mihro, and thanks, George, Tara, and the whole team behind organizing this event. And I'm very happy to, besides helping with the organization, to be also one of the speakers. As I was asked to talk about in our preparation for this event, I will give a historical overview of the ethical deliberations within the Islamic tradition to genomics. This is the outline of my talk today. I will give a brief introduction about how ethical deliberations related to genomics in particular and to bioethical issues in general take place, because they are highly interdisciplinary and a little bit different than how things work in secular bioethics. So I will give a note about this, and then I will speak about two important historical phases in the discussions, Islamic ethical discussions on genomics. One between 90s, roughly 1993 and 2013. This is a phase by itself. And from 1993 to 2013, and then from 2013 till now, where the focus is not anymore on the yes/no question, should we be part of the genomic revolution or not, but we witnessed an important shift from the yes/no question to the how question, how to join, what kind of issues should be addressed, and how these issues should be addressed. So let's start first with the introductory remark about the Islamic bioethical discourse in general. Unlike the situation in Western academies and think tanks, we do not have yet in the Muslim world institutions or institutes or research centers specialized in bioethics. We usually have, for instance, our research center, one of the most advanced attempts in this regard. We have research centers situated within colleges of Islamic studies. Some activities related to hospitals, medical colleges, etc., that you will have discussions about this. But of course, the main contributors to this, we speak about Islamic bioethics, religious discourse, so religious scholars. We expect that the religious scholars would be the ones contributing to this. Yes, that's true. They are the ones contributing to and creating the Islamic bioethical discourse, but they are not doing this alone. Because of their educational background, which is focusing almost exclusively on religious sciences, not on biomedical sciences, on the Arabic language, not other languages, including English or French or German, where updated biomedical information is available. They need we have like a puzzle where not only one person can answer, even if this person is a well-versed religious scholar. So we need collaboration between different groups, not only the group of religious scholars, but others who would help them in these complex issues. So we have two main groups participating in the Islamic bioethical discussions. Besides the conventional group of religious scholars, we also have the biomedical scientists. So we have interdisciplinary discussions. We don't have this yet, this class of bioethicists who already have dual knowledge or interdisciplinary knowledge. This is something still in the making. Hopefully the situation will change soon, inshallah. But so far we have interdisciplinary discussions represented by people from or experts coming from different disciplines and different backgrounds. By the beginning of the 1980s, this interdisciplinary form or way of discussion started to get an institutionalized form. We will have institutions specialized in running Islamic discussions on bioethical issues by combining between or bringing together these biomedical scientists and religious scholars, and sometimes they would add other groups, like for instance those who are well trained in philosophy. Some would add, for instance, those have lawyers and lawmakers, policy makers, etc. But mainly, as I said, were the biomedical scientists and religious scholars. Three main institutions adopted this interdisciplinary approach. The most important, to my mind, the Islamic Organization for Medical Sciences in Kuwait, an organization that intensively collaborates with two others, the Islamic Fiqh Academy in Saudi Arabia, and International Islamic Fiqh Academy also in Saudi Arabia. Most of the time, there is very close coordination between the three institutions. Most of the time, they have also unified positions, but not always. Because it's a matter of ethical deliberations and you have no guarantee whatsoever you will end up having the same opinion. This is how the Islamic bioethical discourse have been working from the 1980s, the institutionalized form of Islamic bioethical discourse. So when we will have the discussions in the starting from the 1990s on genomics in particular, already the discourse and the mechanism of the discourse was already shaped. That we have biomedical scientists working with religious scholars within these institutions, sometimes other institutions that would have occasional contribution to these discussions. We can differentiate, because this is the key issue of the topic, historical overview. We can differentiate between two important historical phases. From 1990, roughly 1993, till 2013, throughout these 2 decades, we have a discussion within the Islamic tradition with one specific focus, one clear main question, which is, we as Muslim world should be part of the incoming or ongoing genomic revolution. Is it wise to do that, to invest huge amounts of money, to be part of this revolution, to contribute to have a role, an important pioneering, contributing role? Or is it better to wait a little bit till we see how things were going, because it's still very much primitive in the beginning. We don't know which direction it will go. We don't know whether it will be good science or bad science, etc. Throughout this period between 1993 and 2013, we had most of the discussions resolved on this big question. Of course, we have so much sub-questions, but this was the key idea. The three institutions that I spoke about dedicated a good number of their activities and meetings. In our survey, we could identify at least 15 meetings, conferences, workshops, etc. Organized, where both biomedical scientists and religious scholars deliberated and debated on these issues. A great deal, and the most important, most influential discussions on this issue were hosted by these three institutions. I use here the timeline of the history of genetics and genomics. This is the timeline provided by the National Institute of Genomic Research. As you see here, it started in 1990. This was launching the Human Genome Project. I can say that since then also the discussions in the Muslim world started, and in the Islamic discourse started. This was a global initiative. A very important project, the most important biomedical venture in our modern time, as it was described by the people who participated in the ethical deliberations within the Islamic tradition. And since the beginning of the 90s, we will have important meetings. One of the earliest that I have seen was this one, Ethical Implications of Modern Research in Genetics, organized in 1993 by the College of Science here at Qatar University. And proceedings of this conference or the seminar were published both in English and in Arabic. So this is an important landmark in the discussions, 1993, just 3 years after the Human Genome Project. 1998, we have a very important and seminal publication coming from a seminar organized by the aforementioned Islamic Organization for Medical Sciences. They held it in 1998, and they published it twice. The second edition was It was published in 1998. In 2006, we have here a repetition of the discussions, but the scope was broadened to include also secular and other religious perspectives, not only the Islamic perspectives. And since then, so many discussions and meetings and conferences, as I told you, about 15 at least meetings we have traced. Outside all the disagreements that we will speak about in a second, there was a general agreement about 2 points. The one is that the world is on the eve of the biggest scientific revolution ever. That's the Human Genome Project, the Genomic Revolution, etc. This is on one hand. On the other hand, there is a deplorable state in the Muslim world of scientific research in general. And we speak here about the 90s and genomics in particular, or genetics and genomics in particular. So something big is coming to our world, and we lag much behind what's happening at the moment. The other agreed upon fact is about the significance of the Human Genome Project, which was launched in 1990. We see this statement in many, in the recommendations of many of the meetings that I refer to, these 15 or plus meetings. They said the current effort to map and sequence the entire human genome, this is in reference, direct reference to the Human Genome Project, is without doubt the most significant and ambitious undertaking of biological research in modern times. So something very big is going on in the United States in collaboration with other countries around the world. So what should we do? I cannot give a detailed analysis of what happened throughout these 2 decades, but we can differentiate between 2 main positions. One position, I call it the cautious approach. I'm very timid to say conservative or anti-genomic approach, because this was not the case. It's not accurate. But the cautious approach, want to say that maybe it's good, this genomic revolution, but also maybe it's bad. So we are not in a position to judge now. It's the no position. We call it in Islamic law at-tawaqquf. Sometimes the religious scholar, when you are quite insecure and unsure about the good and the bad in the issue at hand, you say, I have no position and I do tawaqquf. This is very close to this, the cautious approach. So they were more inclined to say, let the Western world try this, try the waters, the difficult waters of genomics, and then we will see if it turned up something good, we can use it, otherwise we don't. 3 main issues, and the accommodationist approach, were very much pro adopting the genomic revolution, being part of it, contributing to it, and whenever necessary also ethicizing this genomic revolution, streamlining it in order to go into the main direction. 3 main points were discussed by these 2 approaches, we can say. One of them is about the relationship between science and religion. The scholars and the scientists who sided with the cautious approach found that modern science is completely divorced from religion, or irrevocably divorced from religion. Scientists are not religious people. Many of them are not believers, even not speaking about Muslims. And even if they happen to be religious people or believers or Muslims, this religious moral world does not make part of what they are doing in science. So they let their religion outside the lab. And when they get into the lab, all what they care about is achieving results, getting funds for their projects. This is what they care about. And he said that we have different examples throughout history where science was misused by scientists. Why? Because these people are not afraid of God. They are not governed by religious ethics. And we have the notorious example of nuclear energy. In the 1990s, when they were discussing this, the name of the US Department of Energy was linked, of course, it was linked to the Manhattan Project that was responsible for the nuclear bomb later on. And the US Department of Energy was also involved in the Human Genome Project. So they said it's not very far that things can go bad. So this was one concern. The other concern was about the higher objectives of Sharia, and this has to do with Islamic studies. So I need to explain a little bit. For a religious scholar, if I want to judge a specific act, behavior, or an issue at hand, I look into the text, scriptural references in the Quran, the Sunnah, etc., in order to come to a conclusion. If I don't have this, and this is the case with many bioethical issues, more particularly genomics, then I look into, I try to approach this through the lens of the so-called higher objectives of Sharia. Sharia, or the religio-ethical system revealed by God to humans in order to achieve a specific objectives. It's a teleological discussion, thinking about the telos, the objectives. And they said that one of the objectives, of the higher objectives of Sharia, is protecting the offspring, protecting the future generations through human reproduction, etc. And they said we can situate the genomics within this higher objective of Sharia. They are meant in order to protect the future generations. If this is the case, if it is about human reproduction, then it must be about marriage, then it must be about relationship between man and woman, etc. This is the context. And within this particular context, we cannot say everything is permissible unless we have a reason to call it otherwise or to judge it otherwise. It is the opposite. The rule should be the opposite. For instance, in Islam, I cannot have a relationship with a woman to whom I am not related through marriage. Why? Because everything is every woman is prohibited to have relationship with, unless you are married. So we say, by extension, everything in genomics should be judged as prohibited, or at least stopped. We cannot judge it as good unless we have a good reason and a good reasoning process, after which we say that it is good. So there is original prohibition for this. And he said, additionally, considering that the Muslims lag behind this scientific revolution, actually we will have no role for this. So the Western academies, these scientists whose science is divorced from religion, will decide what they think is good. And we will not be part of this. We will be just consumers. So it's better to wait till we have better vision about how things will go. This was a group thinking this way and adopting this position. They were, of course, numerically speaking, in the minority position, but they were there and they were important names in this. The other approach, which was the majority approach, clearly supported by a great deal of scientists and also religious scholars, they were much more accommodationist. First of all, they said it's not true that science is divorced from religion all the way. And if we have the notorious example of nuclear energy, we have the good example of Francis Collins. Francis Collins, his name was mentioned more than once in these discussions as a very good example, linking in his works. Later, he also published his book, Language of God, showing that his work on genomics strengthened his religious beliefs and his belief in God, etc. So they said it's not true that modern science, more particularly genomics, is necessarily irrevocably divorced from religion. They also added that this supposed divorce between science and religion does not necessarily mean divorce between science and ethics. So scientists are bound by ethics and ethical guidelines, even if they are not religious, and even if these ethical guidelines are not called Islamic or Christian or Jewish or religious codes of ethics. The second point, when it comes to the higher objectives of Sharia, they disagreed with the first group that we have to judge genomics differently and to say that everything is prohibited unless we have a good reason. They said, no, it's the general rule in Islam is that everything on earth created by God is in principle for us humans to consume, to enjoy the benefits, etc., unless we have a good reason to say that from a religious perspective, this will be prohibited. And they said that, yes, genomics can entail some harms, like any other scientific venture, like any other human activity. But the issue is how to weigh these harms against the actual or expected benefits. And by way of weighing these two, they said, so far we can safely say that engaging in genomics will yield more benefits than harms. And they also did not agree with the idea that Muslims have no role to play. And they said Muslims, not only that they can or they cannot, they have to have a role in this genomic revolution. If they don't anything now, they will not be able to do anything later. So they have to join this genomic revolution as soon as possible. The winning position became clear in 2013 when the International Islamic Fiqh Academy, one of the three institutions I mentioned, in November 2013, they issued a resolution. After all these discussions from 1993 till 2013, and they framed genomics as part of the human centuries-long journey of improving one's knowledge about oneself and one's creator. This is how genomics should be framed. So it is something, yes, it is new, genomics, but it is part of a very old the quest for knowledge, thirst for knowledge. And this is something, they said, which is very much supported, encouraged by Islam. All what we need to care about, they said, is a rigorous risk-benefit assessment of every single application of this ongoing or incoming genomic revolution. And they said that joining the genomic revolution is not an option. Joining the genomic revolution is a duty, and it is a collective duty. So the Muslim world as well collectively should be part of this. Of course, the duty lies first on the rich countries that have the money to fund these very expensive investments, but there is a collective duty on the Muslim world to be part of the genomic revolution and to ethicize it, to streamline it, etc. This was in November 2013. December 2013, the first two Gulf countries launched their national genome projects. Qatar Genome in Qatar here, and the Saudi Human Genome Project, both of them the same month, the same year, December 2013. Not mistaken, at this time it was even the same day. The later other rich Gulf countries also joined. One of the most recent is the Emirati genome program in 2019, and so it started. So the winning approach that there is no problem whatsoever. It's even a religious obligation to be part of this genomic revolution, and then translated into actual nationwide genome projects with huge amounts of huge budgets dedicated to this, and also creating infrastructure. Dr. George Nimmer was, he is situated in precision medicine, the College of Health Sciences, where we have programs on genomics, precision medicine, etc. We have a master program at Qatar University about genetic counseling. So infrastructure is being created. Qatar Biomedical Research Institute also doing a lot of things in this. Sidra Medicine or Sidra Hospital is doing this. Weill Cornell Medicine in Qatar here also are doing many institutes and biomedical institutes are contributing to this in Qatar and outside Qatar. From 2013, and here I will go quite quickly, and then we can end the discussion and go further and deeper into this. The question was about the what question. are we going to handle once we accept to be part of the genomic revolution? So we will have nationwide human genome projects. We will invest intensively and extensively in this kind of research. Then we will see, by extension, specific practical issues that we have to address, what to do with these topics. Our center, the Research Center for Islamic Legislation and Ethics, and also the institutions that I spoke about, we have seen a number of issues very clearly. One of them is about the ethical management of incidental findings. If we are going to sequence people's genomes and we go through these donated genomes and we sequence them, we sometimes come across the so-called incidental results. If we are going to do these genetic tests in the hospitals, the premarital genetic testing, which is now mandatory by law, etc., what to do with the results, especially if they are incidental, not intentionally sought after. We made this study published in both English and Arabic, can be easily downloaded from internet. Very briefly, we said that some of the incidental findings, it's mandatory to reveal and to return back to the people. If these will be life-saving in nature, then you have to, because saving life is mandatory. And some of the incidental findings can be put in the category of prohibited acts, like misattributed paternity. If the incidents relate to, let's say, one child who is not biologically related to the husband, for instance, these cannot be, because we have a specific perception of family and marriage-based family and marriage-based paternity, etc. We detailed this in the study. Another issue was about the genome or genetic editing. I published an article about this in 2019. 2019 also, the International Islamic Academy had a follow-up conference on genomics and spoke about the genome editing. And they said that it's possible, genome editing, under three main conditions: that there is safety and efficacy of the technique followed, and respect for human dignity for people involved in this kind of research or this kind of intervention, and that it should be curative in nature, not for enhancement purposes, for tadawi, not for tahsini. This is one of the key publications we had in the research center. Mihro and some of the participants to this event contributed to this publication, Islamic Ethics and the Genome Question. This is one of the most detailed works on this issue, and the book is available and can be also downloaded for free. It's open access through the publisher Brill. I will not go further through concluding remarks, etc. I will stop here. We can discuss all this later, inshallah, in the Q&A if you have any questions about my talk. Thank you so much.
Thank you so much, Dr. Ghali. That's an incredibly expansive overview of the history of the interdisciplinary dialogue and nature of the deliberations between scholars and scientists. There's a question that has come in, which I'll start with, Dr. Ghali, and there's a follow-up question that I also have. You've talked about these institutionally based religious initiatives to arrive at decisions around what the Islamic perspectives are on genomic technologies, but how might these institutionally based religious engagements with genetics and genomics that have taken place amongst Muslim scholars and biomedical scientists evolve over the coming years and decades? Do you envisage new spaces or ways in which these deliberations may take place? And what do you see is the role of the public in these religious deliberations?
Thank you so much. This new form, let's say, of discussions, which we call in Islamic studies collective ijtihad or al-ijtihad al-jama'i. So the religious reasoning is not practiced by religious scholars only, but in collaboration with external experts, if we may say, experts from outside the domain of religious sciences. This form, a specific form of ijtihad, which had its roots in history, as I mentioned in the book on Islamic ethics and the genome question, but was revived particularly to address this. It was because of the public, to start with, because religious scholars used to be those who respond to the questions raised by the public. I have a question as a Muslim, I go to a religious scholar. If I don't know the answer myself, the religious scholar give me the answer, and then I think about it, is this okay? Or I go to another religious scholar and so on. It's as simple as this. But by time people realized, the general public realized, that religious scholars are not well trained enough in order to answer these particular complex bioethical questions that have bioethical nature. So the religious scholars realized that the public are aware of this issue. So in order to gain the public trust, they had to collaborate with people who have a know-how about these complex issues in order to gain the public trust. So the new form of this ijtihad is a response to public concern, so that religious scholars would know the positions that they will adopt will gain the public trust and confidence from the public. So this is a positive part. The negative part in these issues is that in many cases the public can be involved, and their attitude can be quite informative for the participants, whether biomedical scientists or religious scholars, but this does not happen. Why? Because of the nature of the two participants. The geneticists or the biomedical scientists, they have a very special expertise, and most of the time it's a very difficult one to explain. The religious scholars also the same. They have this very special expertise. So they think together we can solve it. There is no need for involving the public. The public, we will help them by providing them the position, the conclusion, what to do as a Muslim individual. Although in many cases, I would say they would have been better served by consulting the public. Like for instance, if we say, look, if there is a public need, then we say it's permissible. If there is no public need, okay, so who will determine there is a public need or not? Ask the public, not myself as a geneticist or myself as a religious scholar. And sometimes they debate together too much in these in their deliberations. One would say there is public need, one would say there is no public need. And there is no need for all this. We can make surveys, we can consult with the people, and then we come to a conclusion. And this can happen even before the conference takes place. So there is positive and there is a negative side in the engagement of the public with these deliberations.
Jazakallah, thank you so much, Dr. Ghali. I mean, that's really, really helpful answer, and it helps to connect with what both George and Tara were saying, about this importance of keeping that public voice and the public input when we're thinking about science, but also ethical deliberations. Dr. Ghali, a question from myself. You mentioned this issue of collective duty or fard kifaya in driving policy making, particularly in the Middle East, and there's been this initiation of population genomic projects that you presented. Is there a role for this collective duty or this notion of fard kifaya within the public space? Is it telling the public that they ought to also have a responsibility to engage with these initiatives? And if so, what is the responsibility for the publics if it's different to the sort of thing that you described at the institution or the national level?
Yes, of course, when these institutions came to the conclusion in 2013 about the collective duty nature of engaging or contributing to the genomic revolution, they were addressing this to the governments. the main addressee, where the governments that they have to dedicate enough budgets for this kind of research. But of course, implementing this needs public support and public contribution. Otherwise, so we have here Qatar Genome program and the Qatar Biobank. If you don't have donations, sample donations from the people, you will not be able to conduct research. So they should give their support, not only in words, but in action as well. What we have seen so far, we have surveys, national surveys here in Qatar. We have a number of studies now about this. The results, provisional results, all speak about very positive attitude, public attitude, about being part of the genomic revolution as a Muslim world and stuff like this. And I think this has to do, yes, with religious sentiments, that there is a public reason believing that, not only among religious scholars and scientists, but the general public, that Islam witnessed a golden age of science during the Islamic civilization. Now this golden age is gone. We can disagree when this was gone, why, but we agree that it is not there at the moment. And that there is an urge, a very strong desire to regain this golden age and to have Islam contributing to this. And they see genomics as one of the great opportunities to do some victories in this regard and to get it back. This creates, contributes to creating a quite positive public attitude towards genomics. I would go even a little bit further, but this is something subjective, to say that the public in the Muslim world are willing to do more concessions in order to facilitate the scientific research and activities of the biomedical scientists and geneticists in the Muslim world than it is the case in the West. Why? Because of this historical background. We were great, now we are not, so we need to regain this golden age again. And how to do this? Give these people the opportunity, the scientists, the opportunity to conduct their research. Second, many religious scholars differentiate between two things: conducting the research, so your theoretical fundamental research, and they say at this level there is nothing that we can say is prohibited. And conducting research in itself is seeking knowledge, exploring new frontiers of human knowledge, and this is always good. There is nothing wrong about this. And they would quote the Quranic verses, وَقُلْ رَبِّ زِدْنِي عِلْمًا, you ask God to increase knowledge and all this stuff. When are we going to interfere as a religious scholar to say stop and we have to judge this good? In the applications. So when it comes to the research, they give open rein to these researchers to do it. But in the West, people are much more critical in this regard. First of all, we do not believe in the West that we have a golden age of science that we lost. We don't have this. We don't have this regret or feeling that we lost an opportunity or something like this. We have even some trends saying that we don't need these scientists and we don't need these specialists. We know better than they are. And anyone writes on the social media what they like. So people are much more critical and they are not willing to give this wide scope of freedom to scientists to do whatever they like. So I would say the public attitude is quite positive in the Muslim world. This is what I believe.
Thank you so much, Dr. Ghali. I mean, there's so many questions that I have around that, about what the ethical issues are in the Muslim world then related to this public motivation around genomic technologies. Is there potentially a danger that the systems within that part of the world will be open to less governance, less scrutiny because of the types of motivations that you're describing?
This is what we will see so far. I think that there is a kind of international borderline that no one can transgress. Because if I'm working in a genome project in Estonia or in Qatar or in Australia, I cannot publish my research if it is not approved, ethically speaking, not only scientifically speaking, and there are international guidelines. Everyone has to stick to this. The other layer that scientists in the Muslim world need to consider are the religious sensitivities, the religious and cultural sensitivities. And from my experience, I'm a member of almost every ethical committee here in Qatar, and always asked to give opinions, etc. From my personal experience, they are very much keen by both Muslims and non-Muslims, religious, non-religious. The scientists are very much keen to make sure that their work will not have backlash from society. So they want to do their work in a way that will be welcomed by the public, that will be will give benefit to the society, and will not harm any of the cultural customs or religious beliefs of the people. This is what I see. There is very good intention. Maybe there are things that I do not see, but I'm speaking about personal, first-hand personal experience. So I would say at the international level, you cannot move around. So you have to stick to the same international guidelines. At the social level, there is concern that they will get backlash from society. How far these things will work out in every single issue, we will see. We have the example, for instance, of the incidental findings, return of incidental findings, if they relate to misattributed paternity. Everywhere, by the way, not only in Muslim context, but everywhere, it's a very sensitive issue that someone will discover that the child is not mine, is not biologically related. So it is a sensitive issue. But when it comes to the Muslim and the Arab world, it's even much more sensitive, I would say. So when the Human Genome Project started, these were one of the very first questions that we received. What should we do in such cases? So that's why this was the first applied research project we had in this regard. And this all because they know, okay, these people, you cannot get children without having, you cannot make family without marriage. So there must be marriage, okay? What happens if within marriage, we say to the people, these are not biologically related. Should we say this? Shouldn't we say this? And when we made this study, and the Qatar Ministry of Health, they formed a committee to discuss the possibility of drafting law related to genomic research. They used the recommendations that we made in this study about the incidental findings, and they adopted our position about the misattributed paternity as part of the incidental findings. This all shows very positive results, I would say, so far.
Thank you so much, Dr. Vali. And I think with that, I'll bring sort of the individual presentations to a close to give us the opportunity as a plenary to discuss some of these issues in more detail. So what I'll do is I'll get Tara and George, if you could put your videos on and also unmute yourselves, that would be fabulous. And what I want to do is give the three of you an opportunity, first of all, if you have any reflections, reactions, any questions you want to be asking of one another, first of all. So yeah, please do go ahead. I do still have a couple of questions that have been submitted that I can then follow up with.
May I step in, because I have to be out at 3, so I'll just have a question for both our panelists in terms of disclosing results in terms of genomics. And I guess this is something that is touchy, whether ethically or pertaining to religious. So when you do a research project and you are doing a genomic testing and you are getting a result, how far can you go disclosing the results to the patient if it is not related to the consent they gave you to do the test for. I'll give you an example. I had a family where you have congenital heart diseases, and we recruited them for that particular purpose, which is to get genomic testing in order to reveal if there is any mutation in their genome that can explain their phenotype. We did got some of the results, but unexpectedly, since we are doing a whole exome sequencing, we got some results for a variant that could lead to sudden death, which is not related to particularly to their condition. They did not, in the consent form, they did not disclose if they want to get any information about the results. They want to get results pertaining to the congenital heart phenotype. So in terms of ethics, in terms of religion, how can you go far holding or disclosing the information?
Excellent question. I'll hand over to Dr. Ghali first of all, and then I'll bring Tara in to provide a perspective of what happens in the UK when these sorts of situations arise.
Yes, thank you. I will try to be as brief as possible because the details are given in the study that we made with the World Innovative Summit for Health with WISH. We said that, first of all, the informed consent should be clear in order to be truly informed that you are consenting for an intervention, in this case testing or genetic testing or scientific research, which is different than the conventional types. Why? Because we can come across findings that you did not intend, we did not intend. So the participants should be aware of this to start with, that the possibility of incidental findings can be there, to get prepared, this one. The second, for the type of incidental findings, what to disclose and what not to disclose. We said that certain incidents must be disclosed. Mainly, very close to what Dr. Nimr mentioned. Something that can lead eventually to life-saving procedures. What would this mean? Not only that we have to make it obligatory, not recommended or advised, obligatory. We discovered a mutation. This mutation can cause death, so it's life-threatening. And we can refer to the participant to a healthcare facility that can treat this. Then it is life-saving procedure, and saving life is a mandatory. Because we have in Islam classical paradigms, like for instance, someone is praying and it is the last time, the last slot in the time allocated for this prayer, and you saw someone drowning in the sea. You can swim and you feel you are confident that you can save the person. Then you have to stop the prayer, which is the first obligation for a Muslim, go to save this person, even if the time slot is not enough to do both. So when you come back, there will be no time to pray on time. So we say you cut it and you do it. So based on this kind of reasoning, we said that then you have to reveal the results. But then it will move from obligatory, let's say, to recommended or permissible or advised, if one of these conditions that I mentioned will not be there. Like for instance, we know the mutation. We know that it can be life-threatening, but there is no facility to treat it now. So I will only give the person the information, but maybe he has to travel, go somewhere else. Maybe he will be able to find it. Maybe it will be covered or not covered. We don't know. So it's like here the person who's seeing someone drowning and you are not confident that you can save. So you only tell the person you are drowning or something. So it will not be that obligatory. Or we know the mutation and we are not sure if it is life-threatening or not. Maybe there are epigenetic factors, for instance. So we say here to the researchers, you can do that, but you don't have to. You have to only if it is life-saving. It will lead to the possibility of saving the lives of these humans or their future children.
This is very enlightening, because In fact, this is what we have. We have not a documented mutation that is going to be life-threatening, but according to in silico prediction, it could be. Okay, so it is not found elsewhere. It was not published. It was not documented. But what we know about the gene and its function, it might be life-threatening. So I guess I got the answer.
Then it is advised or possible, but not mandatory.
Yeah.
Thank you. Thank you so much, George. Thank you so much, Dr. Ghali. We are fast running out of time, and it just shows why these sessions are so exciting for all of us, because of the types of questions and the types of interactions that it generates. For those of you who are still submitting questions, please do. What I'll do is I'll pose those via email to the speakers and get replies back to you. And please do email us with more questions after the event. I wanted to end by giving Tara, you 30 seconds to reflect on that questions and how it is that those sorts of tensions are dealt with in the UK or in a secular context.
Well, I guess my first reflection is more broadly on the whole webinar, and I guess in some ways there's also, as I expected, about how there's much more in common than there are differences. You know, in terms of different regions, but also different secular versus religious traditions. And obviously, the UK isn't entirely secular. In terms of that specific question, I think I would get to the same point as Dr. Ghali, perhaps for slightly different reasons or similar points. And I think it probably goes back to one thing that says about knowing who you can discuss things with, because sometimes what we feel is we've got the result, we've got to make a decision today about what to do with it. Sometimes pausing and reflecting and knowing who's the expert in this case on sudden death genes, for want of a better term, or what about the ethical consideration. So knowing, having a bit of a network about who you can speak to, because it's not that common that the incidental findings are absolutely clear cut about what we might all agree as being life-saving. And that's one of the difficulties. And I guess it's that other thing about being upfront with people, that we're looking for an answer to the question you have, and my question is the same as your question. We might not answer that, so we might find something different. And how is that going to be managed? And I think that's a really interesting thing to think about in terms of public discussion and public engagement.
Thank you so much, Tara. And I think that's a fabulous note to end on. You know, the sorts of complementarities there are in different methods of ethical reasoning, and the underlying commitments to both of those is around meeting the sorts of clinical needs and mitigating the sorts of harms that George initially introduced at the start of the seminar. I want to begin the end by thanking the speakers. Thank you so much, George, Tara, and Dr. Ghali for joining us. Thank you so much to Mona for providing technical finesse and expertise for this webinar, and also to my colleague and co-lead of Genome, Arzu Ahmed, who will be chairing the next session. Please do look out for information for that next session, and really look forward to welcoming many of you and more at that session. With that, I will bid you all goodbye and assalamu alaikum.